Pleasure now to turn to the final speaker for our first session, Professor Steve Simpson, who's the Director of the Charles Perkins Centre and an Australian Research Council Laureate Fellow at the University of Sydney. Steve is an amazing scientist. He's not only an amazing scientist, he's an amazing science communicator. And I think many of you have seen and admired his TV film, The Great Southern Land. Steve, it's a pleasure to have you here this morning, and we look forward very much to your talk.
Well, thank you, Suzanne, and what a pleasure it is to be here today. My remit is to cover the entirety of ecology, food and agriculture since 1954. I'm going to go back further than that, however, to the time of settlement in 1788, where Australia comprised a network of countrywide systems of land management, and they were underpinned by a highly sophisticated use of fire management. And under those circumstances, ecology, conservation and food were inextricably linked. If we fast forward to 1954, the birth of the Academy, we had a population of 9 million. We had invasive species, which in addition to Europeans included rabbits, toads, weeds, insects, plants and animal diseases and so forth. And we had a land which was fragmented, suffering wind and water erosion, compaction, salination, and food production was roughly stagnant. Now, it wasn't all bad, however. 1950 had seen Frank Fenner's fantastic work in the release of myxoma against rabbits. And if we look at agricultural production, here's an example with wheat yields. Following a period of declining yield up until around 1900, and then a bit of a resurgence, by 1954, after a period of stagnancy, we were seeing a rise, and that rise was going to lead ultimately to the second part of my talk. But the first part, I'm going to deal with ecology. And indeed, three big questions in ecology. The first one is, why do populations fluctuate or remain stable? The second is, what determines where species exist? And the third is, what determines how many species coexist? In other words, the issue of biodiversity. Now, why on earth should we care about those questions? Well, answers to them are critical to so many aspects of our lives, to conservation planning, to sustainable land, water and marine management, agriculture, food and fisheries, strategies for dealing with climate change, decisions about how big an Australian population we should end up with and how urbanised it should be, and many, many more. So we're going to go back to 1954 and address the first of those questions. And here we saw in that year the beginnings of an Australian debate that actually shaped the field of ecology for decades and indeed still reverberates to this day. And here are the two protagonists, Charles Birch and A.J. Nicholson, two men whose views were very much influenced by the systems they worked on. So if we take Nicholson had a profound belief in the balance of nature. And this is that density-dependent factors control population size. And in his own words, published in 1954, populations are self-governing systems. They regulate their densities in relation to their own properties and those of their environment, by which he really meant food, competitors and natural enemies. Now, Nicholson worked on blowflies, amongst other things, and in particular on what's become known as the Australian sheep blowfly, which was actually an invasive. It came from South Africa, probably around 1900. And in South Africa, it's a species that invades early carcasses soon after death. It then breeds rapidly and gets out before other flies come in and outcompete it. When it got to Australia, it found that it was outcompeted from the outset, even in fresh carrion. So it moved backwards in the cycle and began to attack living sheep and became one of this country's greatest agricultural challenges. Now, given that Nicholson believed that populations are self-governing, a population that was out of governance, was out of balance, therefore must need some new source of rebalance in the external environment, a biological control agent. And it was that view that played out, particularly through CSIRO, in the establishment of a whole series of biological control programs, including against the sheep blowfly, that were founded on a detailed understanding of the ecology and the physiology of these pest species and their natural enemies. By contrast, if we go back to Charles Birch and his close collaborator, Harold Andrew Arthur, they published The Distribution and Abundance of Animals, a very influential book in the field of ecology, in 1954, and there they put much more emphasis on external processes driving population dynamics in animal and plant species. So things like the weather and other sorts of disturbances, many of which are just random, stochastic, were driving mass changes in population dynamics. And here you can see the reflection of the systems upon which they worked, such as the Australian plague locust, an animal that has extraordinary upsurges in population and then equally rapid crashes, in large part driven by external events, the weather. Now, this idea that stochasticity, chance, the lottery of life, through external agency can really impact population dynamics and community structure has been extremely influential, particularly in Australian marine biology. So, for example, Tony Underwood, in his work on intertidal zones, looking at the stochastic recruitment of larval forms to rock platforms and how that determined the relative abundance of different species. And it's led to the entire Australian coral reef tradition, which is based on this idea of stochastic recruitment, largely explaining differences in diversity and population levels in different places. And this has been a pervasive and important idea, particularly in those marine systems. But like all polarised debates, the one between density dependence and density independence is actually, the truth lies somewhere in the middle. Populations, it transpires, are typically driven by both density-dependent and external independent factors. And even more interestingly, I think, both can produce either randomness in population fluctuations or order. And that was an idea that came to prominence through the work of another extremely eminent Australian ecologist, Bob May. What Bob showed, using simple deterministic population growth models, mathematical models, upon which he founded the whole new field of quantitative ecology in the 1970s, what he showed was that in these models you could have extreme density dependence producing chaotic population fluctuations. So what looks like random external agencies causing populations to go up and down is actually extreme density independence. Now this idea, it's actually very difficult to distinguish between those two alternatives. If you have a randomly fluctuating population, is it density independence or is it density dependence? But the idea that you could encapsulate ecological processes in simple mathematical models turned out to be extremely influential. It's been the foundation of much of disease epidemiology modelling, and it's also made important contributions in relation to issues such as diversity and stability in food webs. On the other hand, external events can also impose order on populations. And that's particularly true in the case of what you might call the predictably unpredictable event in the environment. And Australia typifies that in its ecology through the dynamics driven by fire, flood, and other extreme events. So there's been a whole tradition arising out of Australian ecology where people have looked at the role of the dynamics of, or driving the dynamics of populations of these so-called predictably unpredictable events. Slatcher and Noble's work on dynamics of populations under recurrent fire. Joe Connell, a corresponding fellow from the United States, working here in Australia on coral reefs and rainforest sites, showing that you get maximal diversity when ecological disturbance is not too little and not too great. Mark Westerby and Brian Walker's work on event-driven dynamics in semi-arid and arid zones in Australia. And all of this has shown its mark on the functional design and evolution of our own plants and animals. And we've just heard exactly such a story. Marilyn Renfrey and Hugh Tyndall-Biscoe's work on marsupial reproduction, for example, the work on cooperative breeding in birds, and of course the whole botany of Australia, where we have fire-adapted strategies in our flora. So the second question, why are species where they are? Well, if you think about it, where a particular species is and where it isn't is dependent upon potentially a whole range of things, both biotic and abiotic. And trying to tease these apart is problematic. It's difficult to do following a typical scientific paradigm of manipulation and seeing what happens and trying to establish causal influences. And it was Joe Connell who, and this doesn't sound extraordinary, but turned out to be, in terms of the practice of ecology, he appreciated that you can actually do manipulative field experiments in ecology. They can't be terribly large, but they're very powerful when it comes to teasing apart the factors that control where species exist. And Tony Underwood, again, was one of the great exemplars of the use of this sort of a technology. But you don't have to do manipulative experiments. The observational field study in ecology has turned out to be profoundly influential as well. Nowhere more so, I think, than in Rick Shine's beautiful work on the great toad invasion. Now, as I'm sure we all know, toads were released in Gordon Vale, just south of Cairns in 1935, and have since moved extraordinarily across the country, particularly over the top end. The map here demonstrates not only the extent of that movement, and this is a bit out of date now, they're right over in the Kimberley at the moment, but also the fact, and Rick noticed this, that they were accelerating as they moved westward. So on that invasion front on the westward side, they're now travelling at 50 to 100 kilometres a year, which is pretty quick if you're a small animal like a toad. And what Rick found, as well as demonstrating the ecological impacts, was that what we're seeing here is evolution in ecological time. And it's a really simple principle. The toads at the invasion front, by definition, are the fastest toads. That's why they're there. When it comes to choosing a mate, they only have other fast-moving toads with whom to breed, and they'll yield fast or even faster-moving offspring. It's what Rick calls the Olympic Village effect. So longer-legged invaders actually move faster. Now, manipulative and observational fieldwork at localities still remains the bedrock of ecology, but it has a problem. It doesn't really allow us to scale up to the continental and global scales that we really need if we're to understand something like how to mitigate or understand climate change. In fact, Mark Westerby has written that the lack of experimental evidence about geographical boundaries is now our single biggest weakness for predicting ecological consequences of climate change. That, however, that challenge has been met in the modern computational age. And scaling up through the development of species distribution models is now the most highly cited field in ecology. And that stems back to the work of an Australian biologist and climatologist, Henry Nicks, whose pioneering bioclimate modelling platform has set the scene for all of these models, some of which now are extremely sophisticated and allowing fusion of data that involve field plot, satellite, bioclimatic modelling, and the testing of hypotheses and their alternatives within this modelling framework, and integrating that with field truthing at particular localities. Very powerful approach. And that has led to Australia, actually I would claim, leading the world in applied ecology. We have really some of the most distinguished applied ecologists in the academy, and they collectively combine mathematical modelling, computer simulation, fieldwork and basic biology to provide applied solutions when it comes to how we conserve, restore and manage our resources. Third question, what determines biodiversity? Well, during the 1950s and 60s, there was a largely American-led revolution in the understanding of what determines the numbers of species that are packed into a given piece of ecosystem. It was the idea, Hutchinson's idea, of the niche. So the set of circumstances that are required to sustain a population of a given species. And then the interactions between species determine the numbers that can exist within a given area. But with quantitative ecology and the elegance of very simple mathematical treatments of ecological processes, the organism, in all of its glory and complexity, was relegated to a coefficient in an equation. And then there was a period of pretty much 20 years of so-called trait-free approaches to trying to understand the structure and diversity of species assemblages. And around about that time, I was a young PhD student, travelled from Queensland to London, and I went up after a talk by the very eminent British ecologist George Varley, who'd just given this beautiful lecture about population changes in winter moth in Whiteham Woods near Oxford. And I went up to him trepidatiously and I said, "Oh, Professor Varley, I really enjoyed your talk. I'm working on locust feeding and I want to understand the processes involved in food selection in locusts." And he turned to me and he said, "Minutiae, dear boy, minutiae." And he turned away and spoke to somebody else. And kind of crushing, actually, at the time. And I only wish I could go back and explain to him the impact of those studies at the time, but nevertheless. Happily, and again, this is an Australian revolution, since about the 1990s, there's been a return to the view that we actually need to understand the organism, that traits are more than minutiae if we're going to understand biodiversity. And there's been two complementary traditions that have emerged. One is to look in detail at particular species, and the other is to look across very large numbers of species and try and catalogue functional traits and see whether emerging out of that can be some higher order understanding of how ecosystems and assemblages are constructed. And that, of course, feeds right back to that original tradition, the CSIRO entomology tradition of Nicholson, Waterhouse and others that we began with. And the future of ecology is to take that understanding of the phenotype and to play that out through its impact on population dynamics, in turn on assemblages and biodiversity, ultimately onto ecosystem structures, and then to take account of the abundant feedbacks, ultimately back to the gene via natural selection, and they play out through development and epigenetics to determine the phenotype. That diagram, that heterarchy, defines the future of the discipline. But before you can do any of that, you have to have a decent understanding of your own ecology. And you need a strategy, a plan towards how you marshal your resources towards that ultimate goal. And happily, the Academy there is showing considerable leadership through its National Committee for Ecology, Evolution and Conservation, and the Ecosystem Science Plan is exactly doing that. Which brings me to part two. the Australian agricultural revolution. Now, the Academy has seen some real giants in the study of agriculture and associated studies of plants and animals in agriculture. I could have spent all of my time following multiple different avenues in the second part of this talk. What I've decided to do, and please excuse me for doing so, is taking a case history. That's wheat. And I showed you, I began in the second slide showing how production had fallen, yield had fallen, stagnated, started to rise with the superphosphate and legume revolution. And then since then, there's been quite literally a conservation farming revolution, and yields have risen accordingly. So today, when it comes to wheat, even though this country is dry and fertile, we have saline soils and unsubsidised agriculture, we produce 12% of the world grain trade. How on earth have we managed to do that? Well, it's been thanks to a convergence of a whole series of things, no one thing alone. Political will for natural resource management, that particularly became acute in the 1970s, as did economic drivers, unfavourable terms of trade. We had to get better at producing food under those circumstances. Plant breeders, particularly working with geneticists and enjoying the genetic revolution, have played a critical role, as have agronomists, farmer groups, and the discovery of key enabling technologies. And there's some sort of key overriding messages. Firstly, these successes have arisen due to synergies between genetics and breeding on the one hand and management practices on the other. Neither alone is sufficient. The second is that there's a need to integrate all of these things as a system according to ecological principles, and there's great advances being made. We're not there yet. And finally, both genetics and management have been informed by fundamental science, particularly in relation to new understanding about the physiology of plant growth in our hostile soils, water, phosphorus limited, acid, saline. And Colin Donald, Lloyd Evans and Tony Fisher, if we take water, were the founding fathers of this. And it was beautifully formulated by John Pasteur in 1977 in this formula that yield is a function of water use, the efficiency with which we use water, transpiration efficiency, the efficiency with which the plant uses water, and the efficiency with which we harvest our agricultural produce. And different communities have played a role in each of those: agronomists in water use, physiologists in understanding transpiration efficiency, and breeders when it comes to improving the harvest index of crops. If we take phosphorus, as I said, towards the mid-50s, began the superphosphate revolution, where there was a considerable use of superphosphates to enrich our phosphorus-impoverished environment. We're now taking a different slant, and that is to look for insight at our own native flora, which has evolved to deal with low phosphate habitually in the soil. And in fact, when you add phosphates to the soil, you make the environment toxic for our own native vegetation. A much cleverer solution is to go to our native flora and look for how they manage to deal with low phosphate environments or salinated environments, and to take their physiological tricks and physiological and genetic tricks and to introduce those into our own crop plants. And that's the sort of approach which is starting to bridge the ecological insight and the agricultural necessity. And there are many very distinguished fellows of the Academy involved in that work. Hans Lambers, Marilyn Ball and Rana Munns are among them. We've also needed to gain a new understanding of the fundamental biology and molecular genetics and the pathogenicity of some of the major natural enemies of our own crop plants. And nowhere is that better seen than in the work on cereal rust biology. But we've also had extraordinary advances in key enabling technologies and practices. Herbicides for weed control. Whole new generations of such chemical agents have evolved over the past 40 years. There's been a development of machinery that minimises soil disturbance during sowing of seeds, so reduced tillage, ultimately no-till agriculture. There's been development of machines to handle the residue at harvest and also to incorporate a rotation of crops that can enrich the soil, particularly legumes and canola. And again, that sort of work is underpinned by fundamental research in the case of the legumes into nitrogen fixation and rhizobial associations, learning the tricks that natural selection and evolution has provided over millions of years of evolution in this environment. And what we find now is Australia is one of the greatest across the world adopters of these new practices in agriculture. So if you look at no-till adoption, it's now right up there in the majority of extent of use. 70 to 90 or over 90% adoption of this technology has arisen quite literally over the last 30 years. Precision agriculture through the use of GPS and new sensor technologies has been critical. So we can now have highly targeted spraying, control traffic across land to prevent compaction. So you only drive over particular, really precisely defined runways when moving machinery across crop fields. Inter-row sowing to 2 centimetre resolution, thanks to SAT guidance. And the management of land in a way that takes account of its differences as you move across in soil depth, texture, salinity, elevation, and so forth. And being able to, for example, sense precisely how much of whatever it is that you're dispensing is required there and then, and adjusting its dispensation accordingly. So, for example, fertiliser application. The Green Seeker is a device which senses the greenness as the machine passes over the crop and dispenses fertiliser in proportion to that greenness according to a predetermined algorithm. But that doesn't mean that this new agriculture has been the panacea. It's just allowed other problems to emerge. Stephen Powell's work showing herbicide resistance in weeds and how we might go about mitigating that has been a beautiful example. We've seen multiple herbicide resistance, glyphosate or glyphosate resistance, and we've needed new integrated management solutions. We've seen the emergence of root diseases, leaf diseases, pests that weren't quite such a problem prior to these new agricultural practices. My favourites, locust outbreaks and plagues of mice. If you want to grow locusts and have the perfect conditions for gregarising and swarming, then what you do is no-till agriculture because you don't disturb the egg beds, you provide delimited areas of rich food separated by rangeland that concentrates the animals, gregarises them and turns them into the swarming form. The same has been true of mice. So there are problems. And in fact, this broader issue of fragmentation of native vegetation is one that agriculture and conservation biology have yet to resolve and learn how to deal with. So what's the future? If we take wheat, the future is still bright. The new genetics, both traditional breeding programs interlinked with new genetic technologies, are going to provide, inspired by understanding of our native vegetation, a whole series of new varieties for no-till systems, disease resistance, tolerance to hostile soils, and so forth. The new technologies that are emerging, particularly the robotic technologies, are going to be revolutionary as well. Rapid real-time soil and crop sensing, new unmanned aerial vehicles for crop sensing, robots that can go up and down rows and deliver chemicals as required on a very targeted way, and at a bigger scale, ever-increasing improvements in our ability to forecast weather. These are all technologies that show abundant promise. And of course, there are very many new basic research frontiers, and that's where Australian science will make its big impact. The root-soil interface is one, and another is the idea of starting to take really very seriously the idea that agriculture provides solutions to the epidemic of chronic disease facing this country. So an example here, an early example, is the development of high levels of resistant starch in cereals to help in the fight against bowel cancer, diabetes and heart disease by promoting healthy ecology in the gut. And the whole area of nutraceuticals has a huge way to go and much to offer to agriculture and to ecology. So I predict that we're going to see a return to integrated networks of countrywide systems of land management in which ecology, conservation, food and now health are inextricably linked. And perhaps we will be the biggest estate on earth come the next 60th birthday. Thank you very much, and I would love to say thank you to Mark Westerby and John Kierkegaard, who particularly helped me in areas where I'm not expert, which is the majority of that, and to Rick Shine and TJ Higgins as well for their terrific help and support. And thank you.
So we have time for just a couple of questions. I must say that you've given me a great deal of hope for the future, when sometimes with climate change approaching, I don't feel that hope. But working together across disciplines can make a huge difference. Comments, questions? Yes, Georgia.
Having sat beside TJ Higgins last night, would you like to comment on some of the challenges about getting GM crops into Australia?
Well, the whole GM crop issue is one that I really don't feel particularly able to make a sensible comment on. It does seem to me, however, that by identifying, as we were saying, the key innovations that are found in some of our own native vegetation and using that as a means to enrich our agricultural produce. Now, whether that requires GM technology, it probably does, or whether it can be done through conventional breeding techniques, in part it can be as well. It seems to me that that would provide a context within which I can imagine GM in improvement of crops serving a really important function.
Tony, and I think there was someone with a hand up over there.
I think TJ is much better to answer that question.
Is TJ here? Oh.
Tony Klein, University of Melbourne. Yeah. Isn't there a continuum between classical plant breeding and GM?
Yes.
And it's just a question of techniques rather than fundamental.
I think that's absolutely right, Tony, and that's sort of what I just tried to say.
And it's also a question of communication. I think it was done poorly at the beginning for GM. It was much easier to do in medical. We have another question up the back.
Stuart Turner from the Research School of Earth Sciences, ANU. I'd like to make a comment on your mention of the dismissal of what was called minutiae.
Yes.
I believe that it's terribly important to follow up new ideas in detail, even though to other people they seem insignificant. It's certainly true in my own work, where it's been suggested that I'm doing very small-scale laboratory experiments which have no relevance to the real outside world of oceanography or meteorology or geology. But I believe it's terribly important to understand in detail the physical processes underlying these larger scale problems. And the individuals who are doing the work are the best judges of what's important. And I think this is related to the tendency nowadays to regard big science as being the best science. But unless you have a large group of people working on a problem together, then it's insignificant. You know, you dismiss the little things that are being done. But nevertheless, those small things, as judged by peers who really know about it, is extremely important.
Well, I couldn't agree more. And in fact, if we go back to that comment, those 10 days I spent as a PhD student observing 10 locusts and recording every time they defecated and how many mouthfuls they ate, you can lay claim to an understanding of human obesity, the processes involved in ageing, diabetes, immune function that arose from those experiments. And I'd just love to be able to go back to Vali and say you were wrong. Because you were wrong.
Great. On that note, I do think that we need to move to morning tea. Sorry? I can't hear you. One right at the top because we haven't had one, and then we'll move to morning tea and continue this conversation, which is getting more lively as we get closer, more awake after last night. Yes.
Ian McDougall from ANU. I'm interested in Homo sapiens. Why is there one individual now?
Why is there one individual, sorry?
Well, why is there one species now?
Right.
Why
are we only one species? Right.
And in the past, we had many, many species of hominids or australopithecines, and now we have a dominant Homo sapiens worldwide. Why?
Well,
you never answered this question properly.
I would wager to suggest it was a case of Hutchinsonian competitive exclusion.
In other words, aggression. Certainly that's how I've been reading about it in recent times. We actually wiped out the others.